start by exploring the concept of tonicity, which is crucial for understanding how cells interact with their environment. Tonicity refers to the relative concentration of solutes in a solution outside the cell compared to inside. There are three main types: isotonic, hypotonic, and hypertonic solutions. In an isotonic solution, the concentration of solutes is equal inside and outside the cell, so water moves in and out at the same rate. In a hypotonic solution, the outside has fewer solutes than inside the cell, causing water to enter the cell. Conversely, in a hypertonic solution, the outside has more solutes, so water leaves the cell. These differences in water movement are driven by osmosis and are fundamental to cell survival and function. Now let's connect tonicity to the physical state of plant cells. When a plant cell is placed in a hypotonic solution, water enters the cell, filling the central vacuole and pressing the cell membrane against the cell wall. This state is called turgidity, and the cell is said to be turgid. Turgidity is essential for maintaining the structure and firmness of plants. On the other hand, if the cell is in an isotonic solution, water movement is balanced and the cell becomes flaccid, meaning it is not as firm. Flaccidity can make plants wilt as the cells lose their firmness. These states are directly influenced by the tonicity of the surrounding solution. Let's move to plasmolysis, a process that occurs when a plant cell is placed in a hypertonic solution. Here, water leaves the cell, causing the cell membrane to shrink away from the cell wall. This state is called plasmolyzed. If the cell is returned to a hypotonic solution, water re-enters and the cell can recover, a process known as deplasmolysis. Plasmolysis is often irreversible if prolonged, but short-term exposure can be reversed. These processes are vital for understanding how plant cells respond to their environment and are closely linked to the concepts of turgidity and flaccidity. Sum up: tonicity, turgidity, flaccidity, and plasmolysis are all interconnected. Tonicity determines the direction of water movement, which in turn affects whether a cell becomes turgid, flaccid, or plasmolyzed. In a hypotonic solution, cells become turgid; in isotonic, they are flaccid, and in hypertonic, they undergo plasmolysis. Deplasmolysis can restore a plasmolyzed cell if placed back in a hypotonic solution. Understanding these relationships is essential for grasping how plant cells maintain their shape, function, and overall health in different environments.